Thank you. I didn't know that. I'm glad you did. I can't see behind me. Oh, thank you too. There you go. Okay. There's two steps. There's step one, and that's going to split the water, because I got to use the oxygen. I got to use the hydrogen in step two. So step one, the problem is we discovered step two first. So we have a little bit of naming problems. But right now, it gets got to get down. There are two stages. The first stage has got to have light or it will not work. The second stage, which is not on there yet, does not need light. If it's sunny, that's all right. But light is not required because the light is used to split the water. And once the water is split, the light job is over. And then the second stage uses the hydrogen that's split off and finishes it, so it doesn't need the water. Now, the energy carrier molecules are the ones we're going to be talking about. Just remember NAD and FAD, how they carry things? We've got carriers here too. Now remember now, respiration is a destructive process. It's for your good, but that's a catabolic process. This bad boy is a builder. This is an anabolic process. But they go hand in hand. While this one builds, respiration breaks down. I don't want to say destroy. Destroy has a negative connotation, but you got to destroy it to get back out energy that was trapped by this. Okay? The dark reactions are the ones that they don't happen at night. They just don't need the sun. They happen right now outside there. They're just not using the sun. Okay? And this is where you actually make the carbs. You make the sugar, you make the materials that's going to give more leaves, more roots, out further down. The black eyed pea you eat, that's a seed, of course. Okay? And I need to stress only that I like the term light independent process, but that's a mouthful. But that indicates that you don't need the light. The dark reactions make you think it happens at night, and that's not true. They just don't need light. They can happen in the dark. Okay? So far we have the two steps. The first step needs sunlight to split the water. And those hydrogens are used in this step. Now, let me ask you a question before I go any further. What gas does a plant breathe in? Carbon dioxide. Doesn't really breathe, but you know what I mean. Now, do you remember the three elements that are locked into a molecule of glucose? What are the three elements? Carbon, hydrogen, and oxygen. Okay, the carbon and the oxygen comes from carbon dioxide. The hydrogen is coming from water that was split. So the plant takes in 2/3 of what it needs, the carbon and oxygen, with the gas coming in. But the hydrogen, that comes from the water that was split. And in the dark reactions, they all come together and make that sugar, which contains carbon, hydrogen, oxygen, all bonded together. Okay? So you need to just remember, that's why you water your plants. I tell you earlier, you don't water the leaves, because the leaves don't take the water in, the roots do. I mean, you water, no water, it drips off the ground. You know that, it drips off the ground. But if you want to save time, just take your water hose and put it right there on the pot. Soak in there, and that's how the leaves get it. Sometimes it's my fault that I don't water like I should. And I see them start to droop over, and oh my God! And then I'll just put water right there on the dirt. And within a matter of half an hour, I see them start standing back up. The problem is, though, if you plant in pots, that requires more diligence on your part. I have one plant in a pot by my porch. And I got one plant in the ground by my porch. And one on the ground, I never fool with it. You know why don't you? The ground holds water. But one in the pot, though, And I told my wife, when I get off my lazy butt, I'm going to take that potted plant and I'm going to plant it. So I ain't got water or some of this kind. They keep me good for it to water it by how the leaves are drooping, but that's my problem. So I'll one day get my shovel and dig me a hole and drop it in it. And the one on the ground, I didn't plant that sucker. A seed fell off by itself. I didn't know it was there. And I went, huh, look at that. And it's got the red flowers. Hummingbirds come up, stick the beak in them. I got a hummingbird feeder, and they'll visit both of them about equally. But I like some of the flowers because that's the natural setting. All right. Now, in eukaryotes, that means things with membrane-bound organelles. Those organelles that do this are called chloroplasts. And I showed you earlier how the chloroplast contains these green discs. Each disc is a thylakoid. A bunch of them is a grana. So a grana would be a bigger commodity than a thylakoid. Now, inside the thylakoids is hollow. In any hollow area, anywhere in the world, is a lumen. Even in your straw. I mean, right, if I take it off right here, that passage right there, that's a lumen. And a straw, that where the T comes up, that's a lumen. So lumen is not a word reserved just for this. It's just a word that happens to, oh, that feels good. That feels good. All right? The stroma, now, I have a problem with people confusing stroma with which one? Stroma. Stroma, that's the watery part inside the chloroplast. So what are stomata? You remember what they were? For the guard cells? So what is a stomata? That's where the air goes into the leaf. That's the hole. I wish those wasn't spelled so close. The R makes a difference. And it makes a total difference because they're totally different structures. All right? So this is a picture that I like. It's, of course, some artist drew it, but it really does a good job on it. Light reactions occur in different places. The light reactions, these are green, right? What pigment makes them green? The chlorophyll works called light. This is not green, is it? That's why it doesn't need light. So the water is split by these thylakoids, and the oxygen leaves for me to use, and the hydrogen goes out here for the dark reactions to use. So there's your two-step process. Step one occurs in these green structures, in this granum or this granum, actually in the thylakoid of the granum. Okay? This is a quick overview. This is not the breakdown that you're going to see later on. Okay? And this stands for the light reactions, and this stands for the dark reactions. And the light reactions, like I told you, the hydrogen and the oxygen are separated. And the oxygen leaves, and we use it. The hydrogen, see right there, NADPH, there's your grabber. NADP goes up, empty. The water is split. He grabs the hydrogen and he carries it to this side. Okay? This is P, which is going to be used to add to this. When these two come together, you get that. So the light striking the chlorophyll is doing two things. It's splitting the water to make the H available. It's also making ATP to drive the other cycle. Because something you got to power this thing somehow. What will do by itself? Because can't use sunlight. So what happens? The ATP produced in the light reactions is going to drive the Calvin cycle. Now I said Calvin and Benson, but we always say Calvin cycle. I took this off the internet somewhere, and I just didn't take time to change. I want you to realize it's also called Calvin Benson. But a lot of books will just say Calvin. I know a no name but Calvin. Remember how the citric acid cycle is also called Krebs? I don't know another name for this thing. There might be one, I just don't know it. Okay? So what happens in the light reactions, now phosphorylation actually talks about this happening to make that. That's what's happening. So because phosphorylation, I'm adding a phosphorus to the ADP. That's phosphorylation. And what I get from that is an energized, fully charged molecule of ATP. And now this battery can make this cycle go. Okay? So the light reactions are giving you two things you need. The hydrogen that you're going to have to use to build glucose, and then the power to make it happen. Because light ain't driving this. Light's not driving this one because ATP is. And that's why, if you put a plant in the closet, it's this phase that makes the plant die. It is not that phase. But if you don't have the light, you can't make the ATP. You don't water your plant, you can't make the H's, and it all goes haywire. Okay? So that's just a quick overview, and it's pretty effective. And what comes off the cycle is sugar. And that could be sugar cane, it could be black eyed peas, it could be mashed potatoes, like in the roots. Now, the roots down in the cortex, they're not making anything. Roots can't make anything. Who makes the food for the roots?
The leaves.
The leaves do. And the leaves make the food. Phloem or xylem? Which one carries the food down to the roots? Phloem carries down to the roots. And the roots use it to grow. But there's always more coming in than they need, so they store the rest of it in the cortex. And the potato is bigger and bigger and bigger. And when you cut potato and you get like a baked potato and you cut it, put the butter on top, well, all that white stuff in there, that was the starch that the leaf made, but that root stored. So we eat a lot of roots. Carrots are roots, peanuts. I guess the mashed potatoes and potatoes are a staple of our diet. But the root's not making anything. It is doing mitosis to grow through the soil, but it's not making some food. If you pull all the leaves off, that's why the tree dies. Okay? And right now, on the deciduous tree, the leaves have fallen off probably by now. There's some still falling. I see still leaves fluttering down. The days get shorter, too short for the light reactions to happen. So when the leaves are up there, that's how water gets out. So we got a problem. If the leaves stay on there and they can't make any food, they're going to dry the plant out, and it's going to dehydrate and die. So the natural solution is we'll drop the leaves off in the fall, and they fall off. And you'll find that the dew is not a problem. The dew's problem is in the spring and the summer, when the leaves are actually, it's called transpiration, that they're losing water to the environment. But right now out there, it's raining. It's going to rain today, it said, but because the leaves are gone, there's no draw. A lot of times the ground stays soggy in wintertime because the roots ain't picking. Summertime, good hard rain for long. The ground dries because it's sucking the water out and the roots are using it. Wintertime, I know from hunting deer that the ground stays wet a long time. You have to watch out. Okay? Now, the picture I showed you is saying this. Okay? Now phosphorylation is the addition of phosphate. It's actually a phosphate group, PO4, to such a molecule as AD. You can also add that to AMP, which is adenosine monophosphorus, but that's used in a cell to carry things around. ADP is the dead battery. But when you put on that third phosphate, that dead battery is charged battery. then you can use it. If you just pull off the phosphorus, out comes the energy. Okay? So, phosphorylation of ADP produces ATP only because phosphorylation means put on that third phosphorus. And it works fine. I mean, that's really nice. And the energy that you're saving is trapped in that bond. It's the bond, it's not the phosphorus, it's that bond that has the energy in it. If I pull off the phosphorus, then the bond can bleed out and I can use that energy. Okay? We're going to slow down now. We're going to pick apart the light reactions and make them, they're complicated, but so is glycolysis, but we handle that pretty well. Okay? Light reactions. In the light reactions, there are two photosystems. Photo 1 and photo 2. The light reactions, not dark reactions. So, here's how it works. I ask a question. How many phases are there to photosynthesis? And you would say 2. The light reaction phase, the dark reaction phase. Then I ask, and how many phases are there to the light reaction? And you would say 2. Photosystem 1, then photosystem 2. Okay? So here we go. I just told you that. This is when we messed up. If you look at the order of things, how it really works, photosystem 2 takes place before photosystem 1. And you think, well, what do we call that 1 for? Well, we discovered them backwards. The second one that happens, we knew about that one before we knew about the first one. So we named it photosystem 1 in our ignorance and go, oh my God, this is actually the second one in line. So when we discuss them, I'll always tell you, photosystem 2 happens before photosystem 1 because we discovered them backwards. And we've never changed the names. That would be a bigger quagmire. So the books keep on doing, and they'll tell you, photosystem 2 happens first, and then photo 1 happens second. That's why things appear to be confusing. Just remember, it's only because we discovered the second one first. And we didn't know the second one even existed. I'm getting confused talking about it.
Yeah.
But the whole thing is, question to you, which happens, no, I say, what's the order of occurrence? Photo 1, photo 2, or photo 2, photo 1? That's what you got to remember. The order is backwards. Photo 2 has to take place because it's going to make some stuff that's needed by the other one. Yes?
I understand that part, but then when I read that where it says photo 1, but we discovered photo 1 before we knew that photo 2 existed, isn't that? Right. Is that backwards?
No. If I discovered your daughter was a good singer before I discovered you were, who's oldest, you or your daughter?
Me, because I was here first.
But who did I discover first? The newer one, right?
Yeah.
I know it's confusing because it's me also. Let's just do it. Let me tell you more about it.
Okay.
But just remember
I gotta ignore
that the order is backwards. Just remember that photo number 2 does not happen second in order. It happens first. And photo 1, which makes no sense except for the fact that we discovered it first, happens second in line. So here we go. Both of them use chlorophyll, so that's not the difference. Both happen in the thylakoid membranes. That's not a difference. Now, let me try to explain something that's written on screen. Okay. Let's say you got this primitive organisms, the ones way back in time. They can just have one photosystem. Would it be the first one that happens or the second one that happens? which one would they probably have?
So the question one more time.
Okay, I'm an old bacterium. I don't have both. I don't have 1 and 2. Now, if I'm a really ancient one, I can just have 1. Now remember now, photosystem II happened first. So photo 1 must have evolved from 2. So here I am. I'm a real old organism. My line goes back to the beginning of time. I just have 1 system. Which one would it probably be? Because that was the one that always happens first. I wouldn't have number 2 because it wasn't there yet. How can you have the second one if you never had? Oh my goodness! Let's turn it up. Hang on. I thought I did. What did I do? If I'm a really ancient organism and I only have 1, I have the one that is oldest of the 2, right? Well, the older one of the 2 just happens to be the one that's named Photosystem II. Follow? Okay. That also answers about if you have both of them, which one's got to happen first? Photo 2 must happen first because that is the older of the 2 systems. So if I just have 1, I won't have the second one. I have the first one. Now I won't have the second one. So that's why we say this. Now, because in the prokaryotes, there's no membranes that you can have 2 systems with. In eukaryotes, you have the membranes and you can have both systems in. Now, the prokaryotes, are they big things or are they small things? They're little things. They're like the bacteria, like the yeast. Because you're going to need both of them, like for a big old pine tree out there, or for an oak tree. The big organisms, they need both systems to make enough to get them go. But the little bitty things, I mean, I'm going to go home. I have my bird feeder, my bird bath. I'm going to take some water out of it, and I'm going to look at it on my scope. Because in that water, there's all kinds of little critters you can't see without a scope. But those little critters are probably doing one of them. Now, the algae are doing both of them, because they're not what you call primitive. Okay, so that just explains why prokaryotes only have photo 2. It's the older of the 2 systems. Yeah, eukaryotes, photo 2 happens, and then photo 1 happens. That'd be a discussion question of the first order right there, explaining why a prokaryote will never have photosystem 1, because it's the one that happens second. And the one that happens first is photosystem 2. That's the one they'll have, because you can't have the one that evolved if you ain't got the one that evolved from. Okay. Now, there's 2 kinds of chlorophyll. One operates at radio station 700, and the other happens at radio station. See, these are frequencies. When you tune your radio, you're just changing frequencies. You tune in new stations. So, photo 1 uses the 700 frequency, and photo 2 uses the 680. Now between those 2, which one has the little bit bigger wavelength, a little longer wavelength? Photo, is it 700 or is it 68? That's a little longer. Probably can't tell the difference though. Only 20 numbers apart. Now, both kinds of chlorophyll use electrons that's released because sunlight hits them. Catalyze means what? To put it together, take apart. They take apart water. You give off the oxygen and they get rid of it. They don't want the oxygen, but they save the hydrogen and pass that on to dark reactions, which happen outside in the stroma. Are you with me so far? Mm-hmm. Okay. Now, we're going to discuss them in the order that they were discovered, because it makes more sense, because that's when it stands out pretty easily. However, photo 1 was really discovered second. So we're going to use the 700 to start the process with, okay? Now remember now, photo 1 is happening second line. Okay? Alright. Light of the frequency of 700 strikes that chlorophyll. Now that chlorophyll just answers, it don't do anything to 680. And that makes the electrons move to a higher level. When you heat something, does it expand or does it shrink? It expands. Well, when you put light on something, it also drives electrons out to a bigger level. That makes perfect sense. Now, when you're traveling here, you have one kind of energy. But to get out of here, it takes more to keep you close. Therefore, he has like 10 pounds of energy. He has 20. And that 20 pounds, now when he falls back, then he can let go 10 pounds of energy, right? When he falls back, that 10 pounds can then be used. But I got to kick it out there so it can fall back. So if he's right here going around, he ain't doing me no good. Sunlight strikes this chlorophyll, bam! And when he falls back to where he was, that's when you get the energy. That's pretty cool how nature does that. And it's all again by those electrons. Okay? Ferredoxin is a protein that when they fall back to the original level, releases energy. And that is also an ETC because they fall like down a staircase. You saw one earlier. This is one not just like it, but the same principle. As electrons fall back to where they came from, they give off energy. And the staircase is that ferredoxin, which is the electron transport chain. Now, do you remember back when we discussed oil rig last semester? I know y'all don't, but did you remember what oil rig means though? Do you know right what oil rig stands for?
The oxidation reduction.
Exactly. Oil means oxidation is loss of electrons. Rig means reduction is gain. Now, if I have this thing right here, and he gains in hydrogen, was he oxidized or reduced?
Reduced.
That makes no sense at all, does it? If something goes bigger, I'm saying he got bigger. He wasn't reduced in size, but see, those words aren't about how big they are. It's about a chemical reaction. So oil rig is what my clue is. OIL, oxidation is loss. Rig, reduction is gain. And that's why I said this energy is used to reduce this. And I don't mean make it smaller, I mean tack on the edge. Because you gain something, right? That's reduction. And to me, that still makes no sense at all. But if I reduce my weight, I don't gain 50 pounds. But that's not why the chemist uses it. And I've never liked that. I understand it kind of because of the oil rig. But that's the oil rig that'll help her, I don't know what I do. I'd be lost. Okay? Now, remember now the H's that you got when you split the water? Those are inside, they're just kind of floating around the thylakoid. They got to get outside. They got to get outside because they got to be used in the stroma, not inside that green circle. They got to be outside in the yellow area to be used. And then when they get outside there, they can be used to make sugar. The Calvin cycle does that. Now, you know NADP is a carrier, right? So that thing was reduced, and when reduced, it became this. Okay? Now, remember that electron that's falling down the stairway in the electron transport chain? That also releases energy that drives the Calvin cycle, along with ATPs that are coming from the light reactions. So the Calvin cycle which is a big machine, has two sources of energy, ATP and the ETC, the electron transport chain. Now you got it's a lot simpler splitting water than then building that big old carbohydrate. So this slide lost you? Are you with me so far?
Yes.
Okay, and I'll review now and then, but if you ever fall off the wagon, let me know. I'll come back and get you. Okay. This is a picture showing you an artist rendition of Photosystem I, Photo 1. Now, one more time. Is this happening first or second in order?
Second.
Actually second. Actually second. And I'll show you how it all works. Okay. So light strikes this chlorophyll, and only light in the range of 700 nanometers can make this work. All right. When light strikes it, it sends off electrons. The electrons move to a higher level. Now they contain energy I can use. And as electrons fall down this chain through ferredoxin, they're making the energy. And now the energy would then take these two H's and tack them on there and make that. These won't tack on without the energy to make the bond form. Remember ADP making ATP? Gotta have the energy to make the bond. So to make this thing bond, I need a supply of energy. And there it comes. So they're both working together. They're not separate things, but they're both working hand in hand. The NADP has got to carry that hydrogen outside thylakoid to where it's going to be used in the stroma. But the problem is, how do I make the ADP grab hold to it? Well, the electron transport chain provides energy to grab hold to it. So, but it all starts with what? And without light, then nothing's going to happen. Okay? So that's photo 1. Now, let me show you something. This electron is gone. If you run your car and you never stop to gas it up, what's going to happen to you? Well, the thing here, he's always losing electrons. He never gasses up. He can't do that. You follow me? He will give out, it'll grind to a halt. So begin to, they say, I wonder what's giving him the electrons. Then we discovered the other photosystem. Remember? I mean, if you buy a brand new car and it's empty, what's the first thing you must do to drive around Columbus? You got to put gas in. And as you drive it, it goes empty, right? And you must do what? Well, here's your brand new car. He's using gas. I see no gas being put back in that tank. Those electrons, they're just leaving. Then they'll get put back. And then some smart guy goes, there's got to be something happening to put those back so they can keep being traveled out. That's when they discovered the photosystem. And they called it number 2. But actually it happens first. Because if you don't have the electrons, can you do this? So, it's going to confuse you. Photosystem II has to happen first to gas up photosystem I. Because if photosystem II never happens, what's going to happen to photo 1? It's going to give out a gas. So someone smarter than me was seeing all the electrons being depleted, and he thought, well, how are they getting back? You can't make electrons. How are they getting back? And that's when they discovered the other photosystem. They went, oh crap, we named them backwards. But this is the one easy to find. Okay, so we begin to wonder, like I tell you, if he's always losing electrons, why doesn't he run out and everything grind to a halt like a car would? There must be something more going on. Because something is replacing the electrons he's losing. We didn't know what something was. So we began to wonder about who was supplying the electrons to photosystem I. And we discovered it, and we named that bad boy Photosystem II, knowing full well it's got to happen first. You got to gas the car before you take off on a ride. Okay? Now there's another chlorophyll that has the 680 as its favorite light. And it's the one actually splitting the water. It's the one splitting the water. Not the other folks. This is the one that's going to split the water. 'Cause you split the water first. And those electrons are given to photosystem, the other photosystem. That's how you get the electrons. So you split the water and you get these two products. And of course this one's going to be given off. But you also give up electrons when you split that water. Now those electrons go to a higher energy level. And they also travel down their own electron transport chain. I'll show you a picture in a minute. And their chain is not like the one in photo 1. Come on, Hugo. Okay. And you're going to be totally lost.
It's okay.
All right. Okay. So far we have photosystem 2 happening before photosystem 1, because the electrons coming from 1 have to be supplied by photosystem 2. And photosystem 2 uses the 680 wavelength. Photo 1 uses it's the same color. You wouldn't know the difference in shade. Not by 20 nanometers. All right. The electrons from the water travel down the photo 2 electron transport chain, and then they're used to phosphorylate again and make ATP. And those ATPs are used to drive the dark reactions. The dark reactions don't need sunlight because they have the ATP. And my picture is going to clear it all up, I hope. Now, you remember that photo 1 used ferredoxin as a stairway? Photo 2 uses 4 compounds. And I'm never going to make you know those names. And these are the 4 that build this ETC. That's a bit more complex, isn't it? Now using 1, I can do it, but using 4, good Lord, what a complicated process. But this does form a more efficient transport chain. If you're going to use a stairway to make energy, and you have 3 steps versus 10 steps, which one's going to make more energy? The 10-stepper. This is a 10-stepper. Photosystem 1 uses the 3-stepper. It makes energy, but not like this bad boy does. Okay? If you look, see plastocyanin? That's the last carrier in this complicated chain. And when the electrons leave him, they go and fill the gas tank up of photosystem 1. When those electrons are used up by those 4 carriers, when they get done, they're going where they need it the most, to photosystem 1, so they can do it again. So the electrons that you see in photosystem 1 actually came from where?
Photosystem 2.
The second one, the one that happened first. And if that one stops happening and you stop providing electrons, everything grinds to a halt. Because the gas station's closed and you can't regas. So one more time now, in the order of things happening, now you know that photosystem 2 has to happen first because it's going to provide the electron that's going to drive photosystem 1. Even though it's misnamed, that's the order, it's got to be that way. Now, this is, and I did a lot of work on this slide to make it work. And I hope you understand it, so let's try it. Okay. You water your plant, right? Okay. This light during this system will split this water. And when you split the water, you get 3 things. I get electrons, I get hydrogen, I get oxygen. You with me so far? Okay. Now, the oxygen is released into the air, and I can breathe it and keep me alive. Okay. The hydrogen has got to be bonded to a carrier, the NADP, right? Now, where's the energy come from to make that bond happen? The transport chain. Now there's, it goes down this step, down this step, down this step, down this step, and then it goes into, which one is this one you reckon over here? If that's photosystem II, which one is this one?
Photosystem
It's
I.
happening second, right? So the light strikes this, all right? Some things happen. When the light strikes this, water is going to be split. It's going to give off the oxygen because it don't need the oxygen. But the hydrogen's going to be later on strapped right in there. This hydrogen is that hydrogen, and these C's, O's, that's the carbon dioxide that the plant takes in. So here are the ingredients to make glucose. 2 of them come from a gas. This is a gas, but it came from a liquid called water. Okay? Now this O right here is not that O. This O is O of what? Carbon dioxide. This O is the O of
Oxygen.
water. Okay? Now, question. Is that plant making energy by splitting water? Why can't we do it in our cars? We can't.
We don't have the
We
carbohydrates.
can't. Guess why we don't? The oil lobby won't pass the laws. They won't let us do it. Because Exxon and Mobil, if I could put water in my car and gasoline, what happens to their businesses? They go bankrupt. They have paid lobbyists lots of money to make sure that doesn't happen. They got money to spend too. So we know for a fact that plant can use water to make energy, to make machines run. Well, we can use it too, but we can't. One man, one time, either Auburn or Virginia Tech, one of them, he had worked out a little motor, and this motor can make electricity, which can run electric wheels, right? And he put water in his little motor, and then he had something that had to split that water, and the little light glowed. He says, one step further, I can make this into an electric car. Exxon bought that plant and shelved it. They gave him tons of money. And the deal was, now on a small scale this might work. But on a big scale this might not work. But we're scared. We're going to pay you right now to stop your research, sell me your rights, and forget about it. What would you have done? I'd take your money and run. He did.
I won't.
I would. Because if I'd gone further and it wouldn't work in a car, I've lost.
You lose everything.
So this way, I really wonder if he'd gone further, would it have worked or not? I don't know.
If you had worked, you make a lot of money though.
He'd like made a lot more money. And the question is, a sure thing versus
Unknown.
maybe not. Sure thing. And you can read about that. I mean, there's no real secret. And the oil folks, I guess if I own Exxon, I'd do it too. Protect my livelihood. I mean, I'd do it. And I'd pay people to go to Congress and lobby Congress to not pass laws making it possible. I mean, I know I'd do it. It's all money. But we know the plant, we know it works because plants do it. Plants do it free of charge. All right, now, so, one more time. Photosystem II happens, it's got to happen first, doesn't it? I need the electrons to make photosystem I happen. So, light strikes this chlorophyll, and the only light that's going to work is the light in the 680 wavelength. 700 wavelength, nothing happens. But the 680 hits it. I don't know why this ain't 702 like you was. I don't know the significance of having two kind of wavelengths. I have never understood why the good Lord did that. If I get to him, I want to ask him. Why did you do that for? Why wouldn't 700 be both? There's probably a reason, but I don't know. All right, so light strikes this chlorophyll, and then that in turn makes water split into these two gases. This is released, this is captured by NADP and hauled away. The electrons that were released are driven to a higher station, and when they fall back down through this transport chain, they're giving off energy. They're giving off energy, and lots of energy being made. So they're making 4 steps will make 4 times the energy as 1 step, right? Remember ferredoxin? That's a 1 step. So this is making 4 times as much energy as ferret. But then again, well, glycolysis made just a little bit compared to respiration. So we know nature does that, where you have one thing doing 2 and one thing doing 36. You think, why do 2 at all? Because some animals are so little, that's all they can do, that 1. They need that 2. Okay? So the electrons fall down this chain, and I've never asked you, I don't know my step. I just know they're there. I know he's the last one. And I could probably do a PPCP type of a TEF thing, but I just never have cared to know the names of these. I just know there are 4 of them in this chain. I do know the last one is plastocyanin, because that means blue. That means blue. And this wavelength is toward the blue side, as you probably know. The shorter wavelengths go to blue. The longer ones go to red. Okay? Now, the electrons right here, he's not done. He enters photosystem 1. And he goes again. Now, the light reactions are involving those 2 photosystems. One involving the 680 wavelength and one involving 700 wavelength. There's a third thing that's got to happen in using this enzyme. It's called ATP synthase. ATP synthase. You think it's making ATP or breaking it down? It's called synthase. It's making it. Synthesis is making it. Now, that enzyme is also in that membrane that forms the thylakoid. And those H+ ions that you're getting right here, if you don't trap them, they're going to leave like this did. And boom, what would happen then? You couldn't make any food. Okay? So, this is going to be produced in photo 2 when the water is split. And that's going to make ATP synthase produce ATP. And again, this ATP is also used in the dark reaction, which is the Calvin cycle. Okay? And that, of course, makes the sugar, which I call the carbohydrate. It could be fructose, that's a sugar. It could be maltose, that's a sugar. But I just said sugar. I don't want to name glucose as the sugar, because there are other sugars too, but they're all carbohydrates. Okay? Now, there's another picture I found, and I like it. And if you have a camera in color, that's a good way to do it. I found many pictures of this happening, but this is the one that was not terribly busy with all the stuff all over it. They had to do much fixing to it. I had to do some fixing to it to make it teachable. But here's what happened, okay? This chlorophyll is photosystem II. And this chlorophyll is photosystem I. And this light, is this the 680 light or 700? Which one gets the first one? That's the 680. All right, now, when this happens, you know this is being split to make this, right? Okay, this is released. These things, now, this is inside the thylakoid. This is outside. This is inside, because these H's are actually inside. They're not in, they're inside. The electrons travel through the membrane. But now, all these H's, they stay out here. They stay out here. Now, remember that. Now, this electron is going to go through how many carriers right here? 1, 2, 3, or 4? 4 carriers. And when it gets done, the electron enters photosystem 1, right? Now, and what's happening here?
700 light.
All I'm doing is making more. Now, right here, I got some H's inside also. And this is my carrier, because These are not going to the Calvin cycle. Well, they're going in a minute. Let's see what happens. Light strikes this. You split the water. You give off the oxygen. The hydrogen stays out here. Okay? The electron enters photosystem 1. It goes up, and as it goes across, it produces the energy that's going to make this happen. The question is, though, I got to have these inside here to make it happen. Here's how that works. These H's, they're pumped through this enzyme, and they end up being in here. Now, as they pump through there, this energy is being used to add these two together to make what? ATP. Now this one is now free to go over here, and this energy can now make ADP grab him. Now, where's this sucker going to be going? He's already in there. He's in the stroma. He's already in the stroma where he's got to be. So let me tell you again now. This is just first half of this thing. Now, try it again. On my picture here, how many photosystems are working? 2. Which one happens first? Photo 2 or photo 1? Photo 2. Why does that have to go first?
Provide electron.
That provides the electron that's going to drive which one? The other photosystem. If it don't make the electron, this thing, it's going to give out a gas and stop. So 680 hits this one, split the water, get these two products, breathe this one out, this one stays, and this is inside the thylakoid, this is inside the green thylakoid. Now, the electrons, they're going to fall down this chain. They're going to make energy as they fall down that chain. They're going to make ATP, right? Now, then they enter photosystem 1. The one that happens second, but we named the sucker wrong. All right? The electrons will go through here, and all they're going to do is provide the energy to make ADP grab hold of this one. Now, where's this one? Hey, they're all outside here. How does it get inside? Well, that's simple. By the law of diffusion, things move from an area of high concentration to an area of low. What do you see more of the blue H's?
Outside.
Outside. What are they going to do by nature?
Go inside.
What's the only doorway in? What's the only door in? There's your only door in. Can't go in through here. Can't go in through. This is the only way you get in there, and by diffusion, they pass down through there because they're driven by diffusion. Does this require energy? No. It gives off energy. And what you give off forms this bond that makes this phosphate hook to him, and now this ATP can be used to drive the Calvin cycle, which happens in here. And this ATP here, I forgot to come line. I got ATP also happening right here. A lot more than this. Okay? This is making ATP, and so is that. But this slide is actually tracking the H's, not the ATP production. It's designed to track the H's. I might one day go back and put my magic on it and make it track the ATP too. All I gotta do is take this thing right here in that arrow and put it right there, indicating what's happening as he goes to the chain, making ATP. Then I gotta put this thing right here somewhere, indicating that are you making more right here. But I have 4 carriers making a ton of ATP and 1 carrion making a little. All this ATP is being shipped. It's in here. It's in here to be used. I mean, it's already in there. Okay? And all the H's out here, where they come from? The splitting of water. How will they get from the green to the pink? How will they get down there? They got to pass through this like a turnstile. Like you're going to watch wrestling, you go to a turnstile, right? As they pass through there, that thing spins, and that makes the energy that's going to make phosphorylation occur, and that's going to make these two combine, that's phosphorylation, and make that. Now, this thing, you're not through with him yet. This thing is it's got to be caught. The gas will get away now. You got to grab that gas. So this moves over here. And this energy that's being produced by that little single ferredoxin is going to take this and make it become that. And then this is what's going to enter the Calvin cycle and make the sugar. That's just the first half of photosynthesis. That's half they got to have light. You don't have light, that's when, of course, that's when the thing dies. That's why a plant in your closet is not going to live. Okay? This is a summary of what's happening. And this is what you probably know right now. You know that Photosystem II has got to happen first in line to provide the ingredients for Photosystem I to happen next. So you split the water in Photosystem II. The oxygen, the plant gives away. Don't want the oxygen. Wants the hydrogen though. Alright? This hydrogen is attached to NADP. And then that whole thing is sent to Photosystem I. Okay? Now, Photo 1 is the one that has a synthase. Photo 2 doesn't have it. Because the hydrogens that's released in Photosystem 2, they pass through that synthase, which is a part of Photosystem 1. As they pass through there, they produce tons of ATP. That's what they do in making ATP. But when the hydrogen's finished, he's not done, is he? He is then trapped by NADP, so he can then be used again in the Calvin cycle to become part of the sugar that you ate. When you eat black-eyed peas, the H part of the P came from water. And the CO part of the P came from carbon dioxide. And when you break it down, the respiration, you put it back where it was. Because you breathe back out the water, and you breathe back out the carbon dioxide, nothing is lost, nothing is gained. It's a recycle. Respiration is the perfect flip-flop to photosynthesis. It's a perfect match. Because what photosynthesis makes, it uses water and carbon dioxide to make sugar. We take that sugar, we break it down, and we get back the water and the carbon dioxide. And we just keep on cycling. That's why the earth is such a good ecosystem. Animals produce products that plants require, and in return, they pay us back with the food. Perfect. And also, photo sends electrons down this 4-member ETC to make even more ATP for the Calvin cycle. Now, Photo 2 happened first. Photo 1 happened second, as you know now, because we discovered it first. So we named them wrong. All right. Now, the electrons he is using, where did they come from? That first photosystem, which is Photosystem II. Okay? So those electrons are going to be kicked up by what wavelength of light? 680 or 700?
680.
Uh-huh.
700.
Photo 2 was using 680. The second one, Photo 1, is using 700. Okay? And now, the electrons pass down this chain and produce the energy used to trap this H. I need to add one more thing on it too. It looks like I didn't mention the ATP synthase, did I? I'm going to add it here, and I'll add it to your pocket. I'm going to do it right now. I'm going to say 2. Okay, I'm going to say this. The H pluses, those are protons. How do I know that one proton is a hydrogen? That's how big hydrogen. Hydrogen is only one proton. So if I say here's a proton, I can say here's hydrogen. Same thing. Because one proton is hydrogen. So when we discuss these things, okay, well, but why is he hydrogen plus?
Because it's big.
Because I ripped off the electron and sent it somewhere else. He ain't hydrogen nothing, is he? Because if he's hydrogen nothing, he still got the electron. If I rip that sucker off and send the electron down that chain, he has to become a plus, doesn't he? Because now that proton has no member. So H plus is an ion, and the electron that used to be here is now falling down their stairway. And that's why they always come together. The electron and this, because the electron came from this. When it comes off of it, then the H, which was 0, becomes H, which is a plus. And that H plus can be used then to make sugar, and that electron can be used to make ATP to drive the other processes. It's a beautiful system. When you start to really understand it, you go, man, that's so cool. And it's just so, biochemistry, when you get past all the, I'm scared of it, it's really quite amazing. All right? So the H pluses in the lumen of the thylakoid pass, I can't say down, because it might be going up too. You just pass through the ATP synthase, and in doing so, phosphorylate. What's that mean? Phosphorylate? To add that phosphate. Phosphorylate ADP into what? ATP. Which is used to drive which cycle? What's happening inside the stroma? What's that guy's name?
Calvin.
The Calvin cycle. I'll make my PowerPoints. I think that's how I do it. I ain't got a book in front of me. And my wife goes, "You're smarter than I thought you were." I said, "Well, I married you, so I'm pretty smart." But, she sees me make these things. I got no books out, and I'm just, but the pictures, see, those pictures, I read them like a map. And I don't need words of the picture. And I find 4 or 5 pictures, and I see what's happening here. To me, a picture really is worth 1,000 words.
A million one.
And now, let me line these up so that I hope I don't.
You left out an O in phosphorylation.
Oh, it even says so. Let's see if he's going to correct that for me. No, it ain't going to correct that. Too big a word. It ain't going to correct it. Sometimes they'll correct it. But now it's gone. So it knows the word. Why didn't it tell me the word? Now, it will never know this in here. So what I can do is add it to my dictionary. So if I ever use it again, it won't stop me. But now it's back again. So I got to add it to my dictionary. So now watch out though. Okay, that thing is misspelled when you add it. You added something wrong to your dictionary. So be sure you check it before you add it.
Probably because there's no space after the ATP.
Where?
The ATP, there's no space. It's looking at it as one word. That's probably.
I bet you're right. It's not saying ATP. It's saying ATP. It's in space. Probably won't put space in there. Probably. Because we don't want that. I do want to put a dash. I want to maybe use up some of that line, so I can do that. And I don't want to make a new slide to finish this. So my goal is to try to keep it on one slide. It ain't gonna work, is it? I'm gonna back and back off like that and just leave it like that. That way you'll follow me, right? Because I don't want that to come and make a new slide. And I like the size of the font. But 36 is a bit big. Let me make that 32. Then I can probably do all one while I sit down at 32. Now, thylakoid. And I've got to take that back so it will do it. Then I put a return here, come across. And I might be able to spell syntax now. Yeah. And then I come over here and I return. So I hope you have what I just did, that you copied it as I worked through it. Okay, that's what I told you. I mean, it's what I told you, and it should be obvious. All right. I don't even want to start this. One thing, time is up for the lecture anyway. We'll be back here for that. I need a big screen. So I'll see you back here at 2:30, and we'll come back on next Wednesday. We'll start here. I hope you haven't forgotten the light reactions in a week.